My name is Kelly Hunter. I'm the Chief Scientific Officer here at Propath, and I specialise in deploying spatial technologies in biomedical research and translational research. We were motivated to partner with Lunaphore because we were looking for a technology to fit into our workflow to give us the ability to customise proteomics panels, particularly for translational research and clinical trial assays. I think it's really important, particularly in the clinical trial space where there is very limited samples available for the research endpoints. We saw clients really benefiting from extracting the most amount of data they could from single sections, and COMET really suited our requirement there. When we were approached by Lunaphore to test a cyclic integrated imaging platform with this staining technology at its core, I was really excited. Integrating the fast fluidics exchange staining technology into a cyclic imaging platform comes with a number of benefits for us. The first is just a really quick throughput. Each staining round is dramatically reduced, and this is something that we saw from the LabSat. An extension of this benefit is a real shortcut of the panel development time. Again, this is something that we saw in our collaboration with Lunaphore and LabSat. The time to identifying, optimizing and finalizing antibodies and panels is dramatically reduced. The quicker we get our panels developed, the quicker we can get to generating the real data. The combination of the automation offered by the integrated staining and imaging on the COMET platform, combined with the high throughput of the fast fluidics exchange staining technology, provides the scalability and quality required for biopharma studies and clinical trials. It's really important in clinical studies to have flexibility in panel design, whether it's the ability to include diagnostic antibodies or antibodies used elsewhere, and to be able to pivot between clones when they're not providing the specificity or sensitivity required. COMET really enables this flexibility by being able to drop off-the-shelf antibodies directly onto the platform and amplify them. The multiomics capability on COMET enables us to address tricky targets or trace RNA therapeutics or answer additional questions on the same slide. The reproducibility we've demonstrated, not just between slides or between runs on COMET, but also between our multiple instruments, enable large-scale and long-running studies. The customization available on COMET is good for us to be able to drop in biomarkers and also therapeutics and antibody therapeutics. We can trace them on the platform at the same time as being able to measure the cellular response. But within that question, you've got room. Typically, if you were to have a smaller 5 or 6 plex panel, you'd have a very focused question. What this enables you to do is to take those questions and then explore a bit around them, dive deep into individual cell compartments or add in certain pathways to help understand the response to therapies. And once you understand that mechanism, you can trace not just the biomarker, but also the response. There might be some prerequisites. You might need a certain cell type present or a certain activation state, and you can trace those into other indications where your biomarker might be present. You do a tissue screen at single plex IHC, find your biomarkers, but then you can use COMET to really understand if the environment required for the response that you need is present in other indications to help you repurpose. COMET is an invaluable tool during clinical trials for taking the limited amount of tissue available for research outputs and extracting as much information as possible on highly focused, customized panels to really pick apart the cellular response to therapy, while we still regularly develop panels with a high proportion of custom markers on COMET because it's very well suited to this application. Over the last few years, we've built up a large library of optimized markers, but also fully optimized panels. COMET's really good for recombining elements of those panels and shortcutting the development for future studies. Pharma studies require us to be able to measure the analytes that are required on the samples that are available. These samples are very often standard clinical FFP samples, and they're often very limited. COMET enables us to generate custom panels to analyze these samples, and through a combination of multiomics on instrument, being able to detect RNA and protein, COMET enables us to measure what we need to measure with the samples that we have. Together with technology partners like Lunaphore, we provide an end-to-end service from tissue processing to data analysis, offering access to a suite of complementary spatial technologies to accelerate biomarker discovery and drug development programs. Over the past few years, we've worked with Lunaphore to develop workflows, test new methodologies, and really establish this platform in biopharma research. Over the coming year or two, I'm really looking forward to continuing that partnership. Innovation is a key part of our relationship. I'm looking forward to further developing those workflows, maturing the platform, and integrating COMET more deeply with the other platforms we run here at Propath, and continue to embed the application of COMET in biopharma research and clinical trial delivery. Obviously, we have an excellent team here at Propath. We have a range of functions from the scientists who are experts on running these platforms and the technicians that help run the lab with them. We have postdoc study managers who have a range of biological expertise as well as the expertise to combine these platforms and these data sets. What we like most about COMET here is the flexibility of the platform, the ability to buy off-the-shelf antibodies, and the day they arrive in the lab to fully characterize them and to understand whether they're going to work, whether they're going to provide value in a panel. The flexibility to rebuild panels, to add custom reagents, the multiomic capability, and how gentle it is with the tissue, so we can, at least perform H&E on the slide afterwards, if not other modes such as high parameter spatial transcriptomics. It's really important for us, the automation, the inbuilt automation, because we're running large slide numbers over long periods of time, and removing that variability from manual interactions. The COMET service is extremely popular. It's extremely well received by our clients, and we do everything from long planned clinical trials designed well before the samples have been collected to last minute small targeted panels developed to answer specific questions live during clinical trial analyses from other platforms. And there's no other platform we have that can respond so quickly to customers' needs, adjusting the panel contents or building new panels when new questions arrive live during the study. Panels are important. Commercial panels available to answer the standard questions, broad immune compartments, the classic tumor markers and checkpoint markers are going to be popular with a lot of people. Our clients always have custom requirements. COMET is uniquely well prepared for handling those customizations, whether that's taking existing panels and adding to them or building de novo 30-plex panels, which we do with clients. I think the flexibility to be able to drop in and drop out, to recombine, and to be able to trace the biomarker that the client has, rather than be restricted by the limitations of the platform or the samples. Particularly when talking about modern biological therapeutics, be it immunotherapies or siRNAs, viral delivered therapies, COMET offers the tools to trace those in the context of the cellular response. In the age of big data, it's really important not to be compounding the problem and generating more data sets. We built a complete, fully validated image and data analysis pipeline to deliver end-to-end insights on clinical trial samples. I think it is being used routinely in the clinical trial space. And I think the adoption of high-plex MIF has been rapid, driven by the sheer value of the data that comes out of it. Once it was shown to be possible to generate this sort of data in this sort of environment, it gets very difficult to not generate that data. It's difficult because IHC has been a standard for so long, since the 50s, and nothing's changed ever since. It has been very difficult to convince people to move from chromogenic to fluorescence, and low-plex, multiplex fluorescence wasn't enough to make that tip. The high-plex and high throughput is the incentive to move over. It's better for data analysis as well. For us, chromogenic, particularly multiplex chromogenic, is very difficult to quantify reliably, whereas fluorescence is a better data source for for quantifications. There's two things that have made the cut. For years, digital pathology has been struggling to be adopted as well, because it's a change in the way that that data is being handled. There are advantages, although there are disadvantages, because once you standardize something, you don't benefit from the pathologist's wizardry ability to be able to adapt dynamically to the stain quality and to the environment. That's very difficult to capture in an algorithm. The incentive to do digital pathology has been you need to do digital pathology in order to interpret high parameter data sets. So where digital pathology has made some inroads slowly in hospitals for automating IHC quantification and H&Es, mandatory requirement for analysing these data sets. So people are being pulled into using it because they don't have enough tissue, or the question requires multiple analytes in parallel on the same slide, not making inferences across 30 different slides, where you end up being so far away from the block, so far through the block by the end of your 30th section that you can't relate those biomarkers to each other anymore.